And we come to the last speaker of this session from the the Trinity College, Dublin. So the speaker has a typical Irish name. It's Dr. Adriele Prina-Mello. Thank you, Adriele, for coming.
Thank you very much. Can you hear me? Can you? Okay. No. Okay. It's a pleasure, and it's going to be challenging to be the next person after such a full and in-depth talk. I will go back to the in vitro formulation, where there's the need of material science and definition to come to one of my previous colleagues. And in this talk, I will present you about what we have developed in Trinity College as a new formulation for carrier. And through that, we actually tested our 3-tier approach for therapeutics. And I will give you some insight into this. So Trinity is a small university compared to many, but has quite a long historical track record. And recently, we also add ourselves in the position of the global research aspect of nanomedicine, hyperthermia, words that we heard in the past days, iron oxide, and several others that I will go through during my talk. So we tackle and we have approached this aspect while we were discussing today over the fact that we have a personalized medicine approach where it's focused around the treatment, but as we know, the paradigm of treatment, single treatment, doesn't work, so therefore we need to switch towards a more companion diagnostic aspect. And through the interaction with the histopathology of St. James's Hospital, we actually come to discuss with them around the table on how we can best perform what it could be for them, a carrier that they could use for histopathology, but also treatment that can be combined effectively. So through that, we move into the next phase where obviously, yes, we have an emerging need, which is resistance. And this has been discussed from the previous speakers, but also probably by the common aspect that we're through what Jan has actually presented through the resistance through gene response. We need to retarget and retarget continuously the reoccurrence of cancer. So through that, we need to have tools that are actually allowing us to be able to treat this and to deliver the molecular mechanism of the genetic material. So therefore, we see a paradigm shift that in the past has been focused around organs towards the effector of the molecular pathways. And through that, we need to master it, this one, in an efficient way at the size that we know and we are discussing around this conference. So therefore, from that perspective, what we come to the formulation and what is the current treatment in companion diagnostic is the fact that we always have a benefit, but we think an element of risk. So the risk factor then is associated to the intensity of the disease or the pathology that we are curing. And in some cases, just a simple sensitization is not sufficient to accept the benefit. So could this fit into this paradigm? Could the paradigm of nanomedicine fit into the protocol? Well, we've been discussing this one. I've been involved in several controversial discussions, and I learned my lesson. So before I'm going to present our results, I present what the others have done, which means we have carried a very extensive investigation of all the products that have been commercialized or they've been brought into the market through the most convenient administration route or the most efficient administration route. And through that, we have highlighted, we identify the fact that the nano product or the so-called nano product, they're just more soluble and they deliver a higher load. There's nothing more simple than this one, so we're more effective. And that's what we come up with the review that we recently have submitted. Interestingly then, in relation to the membrane and to the structure and the cytoskeletal aspect that is associated with the disease that you're targeting, you can also structure your nanomaterial in a way that you can actually create a more stealthy or a more effective pathway through the internalization process. So there we come to the 3-tier approach that we have formulated. We recently published in Biomaterial, where we actually associate that the tier 1 is you need to design the material to respond to what to many sounds like the obvious route of physicochemical characterization, internalization and mechanism of time dependence, cytotoxicity and biodegradation. If we add the element of the shell, then we know that the biocompatibility is a risk element. When we then move into the theranostic or the therapeutic treatment, we also add the extra layer, which is our beneficial aspect. And if we draw the balance between the two, then we have the two weights to associate to our balance. So therefore, this fits what in toxicology is called the safe by design, and through that we actually went to discuss with the chemistry on this perspective. And we come out with two tools. One is iron oxide with doxorubicin and Nucant. Nucant is a peptide which is synthesized by a partner group with whom we are collaborating, and it works through a receptor, surface receptor, where doxorubicin is a quite known and FDA-approved drugs. The second material, we use gold nanomaterial with the paclitaxel anti-drug cancer, and again, folic acid as a receptor, a peptide receptor, as a receptor for the surface for cancer targeting. Through these two aspects, we've seen that both the two metallic particles have contrast agent responses, and also they are, in a way or another, they actually delivering some response to our measurement. We have looked into that, and as my previous colleagues has presented, size has an importance, and we look into the size of the two materials before you do the shelling. Through the shelling, we went through two formulations where you can actually then achieve the aspect of targeting. So if you don't engineer every aspect and you re-engineer it, you will never achieve the beneficial aspect of it. So we went through, to cut the story very short, we went through it and we actually targeting, and in this case I will show you the projection of the two particles as I show you. One is a 30 nanometer gold iron oxide, gold nanoparticle, which we call nanobox, which is actually hollow and is produced by galvanic replacement, and the second one is a 60 nanometer, same box. As you can see, the internalization is impaired in the smaller, quite surprisingly, compared to the 60 nanometer. And as you can see from the side view of the confocal imaging, you can actually see that the positioning is through the membrane folding, where through this one is actually clearly internalized. Well, not convincingly, we went and we did TEM, and through TEM we actually can see that the nanoparticle 2 has actually reached the liposome, and it can be clearly distinguished by the shape, because they're tetrapods. Tetrapods are quite easy to distinguish, so they are quite clear to see, and through that we can also distinguish also the response of the mitochondria across the aspect of the vacuoles that are generated. So therefore, through this we can actually speculate that this is a true endocytotic response through clathrin-mediated internalization, which is driven by the particle and not by the drugs that is functionalized on the surface. We went looking at the mechanism of internalization, and we can look that in this case is not temperature mediated, so therefore is a transport, is a clear passive transport. So through the sodium azide we don't see any response, but we also see the nanoparticle 2 perform better across the temperature mediated aspect. So there we see the first benefit that brings us to choose nanoparticle 2 to nanoparticle 1. Through this one we went to the biodegradation, which is the endpoint that most of the people are not considering as end of life of our particle. And there we took the two particles, we incubated into cells, which is the cheapest model that we can currently use. We retrieved them after 22, 48, and 72 hours, and you can see how our tetrapods originally imaged at 0 time, they actually change shape. When you then go and do a higher microscopy, which we have done through helium-ion microscopy, and Trinity College is one of the 5 universities that has this facility, you actually see that the shape through time is actually changing, is degrading the material. Okay? If you then look at the variation in size of the particle through time, you can actually see that there is a reduction. So there is a dissolution of the material through the lysosomic response, which goes conversely against the theory that gold can actually be there forever. Through that, we look at the cytotoxicity, and we look at the non-treatment versus the nanoparticle 2 is basically within the biological response, and our positive treatment, which are quantum dots on the same quantum dots, they actually have a positive response through 24 and 72 hours. We look at the biocompatibility, and through that we're looking at the shelling, and the gelatin shell coated that we've been using is actually providing a similar response with the degrade shown at 72 hours by the degradation of the shell. We then went into the targeting, and the molecular paclitaxel actually has a less effective response at 24 and 72 hours than the actual particle fully coated and fully loaded with the paclitaxel. So therefore, the actually targeting through the folate works through the adenocarcinoma cell line model that we're using, which is A549, and through that degrades and deliver the load. This is particularly evident at 72 hours compared to positive control. So I hope through that I've shown you that this could actually be an efficient model if we actually follow the aspect of evaluating every single aspect. But for many that have seen the previous presentation, you say, well, so what? This is an in vitro. Well, through this, we are bringing forward the second formulation that we developed through, and I presented several slides before, through an aspect of teragnosis. And through this one, we are in another European project where the actual particle will actually be used for multimodal approach, will be targeting, hyperthermia treatment, and then delivery. So the obvious candidate are small animals, and through that we have injected them, and we currently are analyzing all the distribution of the particle through, and as you can see here, after immediate 24 hours after injection, you can see that it is accumulating into the lung as part of the circulating distribution. We also are currently doing a 28-day study where we actually deliver an hyperthermia, where we actually co-localize nanoparticle doxorubicin and targeting agent with nucleolin. And I hope to present this one at the next year at Clinam with the full results. The current material has been submitted for publication, and through this I will make some times. I want to acknowledge all the people that were involved into this project, the European and the funding agencies, the people involved throughout the formulation, the design, the biocompatibility, and the tier approach, the imaging facility. And finally, I would like to thank you for your attention. I hope I've catch your interest, but I want to leave you with this final one. So don't forget, 6 o'clock tonight. Thank you very much.
Yeah, very interesting to see whether you will be happy or unhappy this evening.
Tomorrow morning.
So other questions from the audience? I think we can still well have one or two questions.
What is the peptide nucant?
Nucant is part of the nucleolin peptide chain, and it's been formulated by José Corti in Crète in France, and it's part of the Multifarm project which I described, and N6L is one of his formulations that is particularly active. He's published a paper only on the activity of the nucant as a peptide on cancer research, and we've seen that there is an enhancing, this is data that, for the sake of time, there is an enhancing capability when it's actually bound to particles on the activity of internalization through the receptor, and then when there's a secondary drugs or a second active compound, this gets amplified. And this is the point that we are investigating in Multifarm project.
Yeah, please.
Excellent talk. You mentioned hyperthermia. You are using the iron oxide nanoparticles. How do you do the hyperthermia, with magnetic fields, or what is your approach?
We are using an alternating field, yes. We have developed within the Multifarm project an hyperthermia system. It works in, conversely, it's from other systems, it works in the megahertz instead of being in gigahertz. And we deliver in approximately to the surface. I mean, we're talking about surface because it's the easiest. We can monitor it through the magnetic field. We deliver approximately 70 millitesla to the surface. We have tested through IV injection, the localization of the particle, and through intratumoral injection. And this work, as you know, is also carried out by Ingrid Hilger, which is a colleague from Germany, which also is comparing to her system, which is in the gigahertz and has a different field strength.
Okay, I think then we wish to close the session in order to allow also for a short break. Thanks again from my side to all the speakers. And thanks to the audience for joining.